All right, so yeah, I'm Chris. I'm based at the biological station Doñana in Spain. And together with Ivan Gomez-Mestre, I've been looking at genome-sized evolution, as well as some aspects of life history evolution in amphibians. So traditionally, genome size is measured as the C value, which is essentially just the mass in picograms of a haploid nucleus. When people started looking at sort of a broader comparative sort of spread of genome sizes across the tree of life, people very quickly realized that it's actually not very uniform and there's this massive spread. And this was sort of upsetting at first and was sort of deemed the C-value paradox, because you'd find many complex organisms, such as mammals, including humans, having much smaller genomes than a lot of simpler organisms, such as some flatworms or mollusks. Shortly after, so this was in the 1950s, shortly after, people realized that actually most of this variation stems from non-coding parts of the genome. So that was no longer that paradoxical, but somehow the enigma still remained of why is there this massive variation? And moreover, is genome size itself a trait that's under selective pressures? So a lot of work has gone into this, and a lot of people have tried to look at this in different ways, and I'm just going to try and summarize some of these hypotheses in a more simplistic fashion here. So on the one hand, you have these non-adaptive processes. So these are stochastic or selfish, genetic or chromosomal processes that, such as gene duplications, insertions, microsatellites, transposable elements, and these tend to have sort of this overriding effect of increasing the inert genetic content. Studies have shown that genome size do have an effect on properties such as cell replication rate and minimum cell size, and one can imagine that then these properties have direct effects on things like metabolism, developmental rate, growth rate, even egg size. And so if there's selective pressures acting on these life history traits, you kind of expect there to be this feedback into also selecting for smaller genomes. These second group of hypotheses tend to be clustered in adaptive or sometimes nucleotypic DNA hypotheses. And how much either or both of these contribute to the evolution of genome size is still somewhat debated. So we wanted to see, can we find this increase in genome size over time, sort of this unchecked increase, or do we rather find a lot of correlations between life history traits and genome size that might imply this sort of feedback? So we decided to look at amphibians for this particular problem, and the overwhelming reason for that is that they have an absolutely enormous variation in genome size. So the largest of any tetrapods. They happen to also be incredibly diverse in life history. So you can imagine why people started to draw these conclusions, that there might be some link between life history and genome size. And there's also a handful of practical aspects. So we have genome size information for a decent number of species. We've got a phylogeny, and there's also databases on life history traits and geographic distribution that we can use to get some climate data from. So we have 400 species, to be precise, that we can work with, but you'll notice only 3 Sicilians. So that's sort of a major drawback. And so most of the analyses are focusing on frogs and salamanders. But I just wanted to show you what that spread looks like. So this is a phenogram. You have genome size on the y-axis, and time in millions of years before the present on the x-axis. And you don't need me to tell you that these genome sizes aren't uniformly spread. So there's this clear bimodal distributions, with salamanders having absolutely enormous genomes, frogs much smaller genomes. And it looks like Sicilians are somewhere in the middle, and the ancestral trait reconstruction also places the ancestor in the middle, but we definitely need to know more about Sicilians before we can draw any conclusions. So the large salamander genomes, they've been sort of very attractive, and one study in 2012 came to the conclusion that the reason there's these larger genomes is because they have a lower deletion rate. So genomes just accumulate faster than they do in neurons and other vertebrates. But if you look within them, I mean, the spread is more or less the same. So you have comparable variation within both frogs and salamanders. So what might lead to these is still somewhat unclear. So what we wanted to test first is whether these have genome sizes evolved more or less at the same rates for both salamanders or frogs, or whether you've had some shift in rate dynamics that might have led to this divergence. So we used BAM to get rate estimates for this trait for genome size. And very quickly, so what you see here is a heat map overlaid on the phylogeny of the best reconstructed rate parameters for genome size, and hotter colors are faster rates. And what you see is that there was fast rates at the beginning, at the origin of amphibians, but then they cooled over time, but there was no dramatic shift. And salamanders and frogs evolved under the same rate regime. When you look at the time-weighted averages, you again get that impression that, okay, salamanders might have slightly lower rates than frogs, but these distributions are largely overlapping. So there doesn't seem to be this support for this idea that you had this sort of punctuated event that led to, you know, either very large genomes in salamanders or very small genomes in frogs. We did recover one rate shift there, so that's that green dot there. And the Myobatrachoids, it's a superfamily of Australian frogs. They seem to have just increased the rate at which genome size is evolving. And that's largely driven by the fact that you have Pseudophrinae on one of those branches, which has one of the largest genomes of any known frogs, paired together with Platyplectrum, which have much smaller genomes. So it's kind of a disclaimer that Pseudophrinae genome estimate is from one single measurement, so it could be erroneous. But what's interesting is that you also see very differing life history characteristics, such as hatching time and egg size, which I'm going to try and convince you in a moment aren't necessarily trivial when it comes to genome size. So that first idea that I presented was that genomes just accumulate sort of genetic content over time. So if that runaway genome idea was true, what you'd find is that sort of the longer a species was allowed to evolve, the larger its genome would get. And these ideas probably stem from things like the mud puppies. So they have huge genomes, 120 picograms. Just to put that in relation, humans have a genome size of about 3.5 picograms. So really, really big, and you find them sort of at the end of this very isolated branch. So they've had about 100 million years to just evolve independently. So what we did was we took the chronogram and we performed these branch length transformations using the phenotypic rate estimates we got from BAM. So here the branches are corrected for by both time and the rate estimates. And when you plot that against genome size, you do recover this slightly positive relationship in both salamanders and frogs, but they're not significantly different from zero. So we don't find this just all-out increase in genome size over time. So that's really interesting because that kind of opens up the field again for some of these adaptive hypotheses. So again, we're back to this idea that maybe there really is something that's also leading to sort of decreases in genome size. So traditionally, studies have found correlations between environmental traits and also life history and genome size, but they've struggled in trying to put that in a phylogenetic context, as well as disentangling some of the collinearity of traits and also sort of direct and indirect effects. We decided to take a phylogenetic path analysis approach. So this is a sequential equation modeling type of analysis, and it's very hypothesis driven. So instead of just throwing sort of a whole bunch of variables at this, we could try and sort of think about what we're actually expecting to find and whether those models are supported. We decided to go for three sort of base models, three groups of models. So this first group, we decided to look at whether or not there's a direct effect of environment on genome size. So environment affects both life history and genome size independently. We have indirect effect models. So here you have environment affecting life history, and that in turn affects genome size. And then as a third group of model, we've got basically no direct effect of environment. So environments affecting life history, and whatever is causing changes in genome size is then also affecting life history. As parameters for that model, or as variables for those sort of groups of parameters, we took temperature and precipitation. Life history, we looked at hatching time, egg size, and we also included body size to correct for any sort of allometric effects. So these are all traits that you typically come across in the literature looking at amphibian life history and genome size evolution. And then genome size, again, we stuck with our C values from the genome size database. So what we find is that third group of models never comes out on top. So this idea that somehow genome size is also limiting life history doesn't seem to be a very good fit. And both in frogs and salamanders, these first two models, so direct effect of environment or indirect effect of environment, seems to outperform that third group. And when we zoom in, we find that this indirect effect model is the one that came out on top. So basically what that's saying is that in colder, wetter areas, you tend to find species that have longer hatching times. That's also true for species that have larger egg sizes. And it's that hatching time that then, and only that hatching time, that then affects genome size. So this is a sort of scatter plot of just that last relationship there, with the regression coefficients on the arrow and p-values from phylogenetic generalized least squares. So salamanders, much bigger genomes than frogs. Why that is, we still don't know. I'm sure sort of advancements in genomics are going to help with that, but we also really need to find out more about caecilians, in no part to just also understand where this divergence comes from. But it's just interesting to find that the variation within these orders is very comparable. In terms of rates, we didn't uncover, or we didn't discover sort of a shift. So salamanders and frogs seem to be evolving under the same rate dynamic. Maybe there's some exceptions for some parts of that tree, but in general, very uniform. So we can't really propose this idea that some punctual event led to this divergence. There's little support for the runaway genomes hypothesis. I mean, there still is room for that, but what we find is that there's definitely an effect of life history. So you have a minimum hatching period limiting the size of genomes, which is in turn limited by other life history traits as well as the environment. So with that, I want to thank my group, the group I'm working with in Spain, as well as funding sources, and you guys for listening.